So this week we will go through the fatty acid synthesis. Fatty acid synthesis, which I have left last week. I mean, not last week, the previous week. If you remember exactly what we left it. The fatty acid catabolism will involve in 4 steps, right? And the synthesis also involve 4 steps, and that is exactly the reversal of degradation. That is reversal of degradation, fatty acid degradation. Degradation. That's my ink there. So the first step, which involves the degradation, reversal of degradation here. Here the assembling the individual units. That's what we need to do in the synthesis now. Synthesis. I am just recapturing it what you have done before in the synthesis. First step, assembling. Assembling of the individual unit. Okay, individual unit of activated acyl group or acetyl group. I put it acetyl group. Acetyl group. Or acyl group. Activated acyl group. So this is the first step. And the assembling means you have to assemble not one unit, but this is the one unit or the acyl group with assembling with melanyl unit, another fatty acid, melanyl unit, melanyl CoA or melanyl unit. This is the first step, assembling. Meaning you are going to synthesize the fatty acid, remember. So you have acyl group or acetyl group and then melanyl unit. So melanyl unit is a necessary. The first step you need to have the melanyl unit. From this we are going to build or synthesize the fatty acid. Second step, melanyl unit or melanyl CoA condenses to acetyl unit. So example, acetyl unit. 2 plus 2 here, you get 4 carbon fragment. 4 carbon fragment. So now we are synthesizing from a 2 unit to 4 carbon unit. This is the second stage. Okay. Now the third one, next step, the third step, carbonyl group reduced. Carbonyl group reduced. Carbonyl group reduced to methylene group. Methylene group. Carbonyl group means C double bond O. The C double bond O, this is a carbonyl group, will be converted to methylene group, mean CH2. Imagine here you have the carbon 2 valency binding to oxygen. That 2 bond is going to be 2 hydrogens, 2H. So see here the 1 oxygen binding to 1 carbon C double bond O, double bond. So this double bond will become a single bond to attach to 1 hydrogen and another hydrogen, and this is called methylene group. So this is the carbonyl group is reduced to methylene group. That is the step number 3. Okay. And this process of addition of hydrogen, what we normally call it as a reduction. Reduction. Okay. And the following this reduction, number 4th step. Okay. What you get a reduction, you get a dehydration, which involves the methylene group in 3 mini group on that. That is the degradation the step it's exactly the opposite, but here what you get, the CO group is being converted to CH group, and it involves, right, a reduction. That's what I said, reduction. That means addition of hydrogen, reduction, and then it is followed by a dehydration, and then another reduction. So that's what it is involved. Dehydration means water molecule is removed. That means the oxygen is removed as a water molecule, dehydration, and then a reduction, addition of hydrogen, that forms the methylene group. And the fourth one is the formation of, when you add this, the formation of a butyryl coenzyme A. Butyryl coenzyme A. This is the 4-carbon unit which is being formed on that. And then what happened, the next, another activated malonyl-CoA. The butyryl-CoA is the 4-carbon unit. Okay, the butyryl-CoA is the 4-carbon unit, and this butyryl-CoA is now ready to attach to first step with the malonyl coenzyme A. So this is the last step. So this is the 4, another one 2, the another circle, it will go on the 6, meaning 6-carbon unit. So this is the repetition, repeat from step 1. So this process is going on, and then finally what you get, you get a large fatty acid synthesis, large fatty acid molecule synthesis. So this is not one repeat, several repeats, so that you will get carbon skeleton is 18 or carbon 20. You get more and more and more of the carbon chain. Okay. So that is the basic principle which involved in the fatty acid synthesis process, and it also involved the acyl protein, ACP. Have you tried that ACP process which we discussed earlier? I don't know whether we did it or not. Acyl protein. Did we do that? No? Last time? Just we want to have a translocase. There is a specific protein which will bind to the acetyl group. That's called the ACP. That's called the acyl carrier protein. Acyl carrier protein we normally call ACP. What is this acyl? The acyl is nothing but the newly synthesized fatty acid. Fatty acid in the form of newly synthesized fatty acid in the form of acyl coenzyme A. So the acyl coenzyme A, we call it not acetyl. Acyl means you have more than CH3 to CH2, maybe 15 or 16 or 19, any number, and then COO minus. So the acid group. Okay, that's called, we call acyl group. So there is a protein which will carry the acyl carrier protein, and that will be distributed throughout the body. That's what in the function of the acyl carrier protein, or the fatty acid binding protein. So it involves in various metabolism, like a storage of lipid. If the fatty acid is not metabolized, the excess of the fatty acid which is synthesized will be carried out by the acyl carrier protein, and thereby is stored in adipose cells or adipocytes. Okay. Now we have to go into the little of my PowerPoint. Let me grab on that what you. Okay. We discussed about the chlorine ring, where the cobalt, involvement of cobalt and cobalamin and vitamin B12 here, I mean, how they involve in transformation of succinyl-CoA, I mean, propanol-CoA into succinyl-CoA enzyme A. And this is the mechanism of action. And now the change of hydrogen position here and the R over here. Now you can see the homolytic bond cleavage, how the cobalt atom which is present in the middle will change with the cobalamin to formation here. 3+ to 2+ here. And the L-methyl malonyl-CoA that is being converted to succinyl-CoA. I mean, these are the intermediate steps in the mechanism of steric hindrance. And this is another cartoon explaining the same mechanism. And here you can see the acyl coenzyme, acyl-CoA, dehydrogenase, oxidized FAD, here the reduced FAD, FADH2. When the presence of oxygen, hydrogen peroxide is produced, and that's been converted to water in the presence of the catalase, and further oxidation, the mechanism, acetyl-CoA, hydroxy, methyl glutaryl-CoA. These are, all of them are getting on the degradative product of fatty acid. Okay. Finally, you get the beta-hydroxy, 3-hydroxybutyrate and acetone, and they are the ketone bodies. That's what acetoacetate, acetone and hydroxybutyrate, they are the final degradative product of the fatty acids. Or if you have not taken any glucose or carbohydrates or fasting for a long time, your blood level of acetoacetate or hydroxybutyrate and ketone will be increased, that is ketemia, and then if you excrete this in urine because of excess in blood, that can also be transferred to the kidney and filtered, and if you have excess amount, it can also spill into the urine, so you have more of ketone bodies. And the brain tissues can use these ketone bodies as an energy fuel in the brain cells, okay, the neuroglial cells. So that way they can sustain even if you fast for a while, 2-3 days or even 40 days. These acetoacetate and hydroxybutyrate are the stored house of your fatty acid, can act as a fuel molecule for living for a long time. So this is another interesting phenomena. And also, if anybody is fainting sick without eating for 2 days or something, they can easily find out the acetoacetate or acetone or ketone bodies like beta-hydroxybutyric acid. They can present in the blood or in urine. That's another diagnostic test. Also, if untreated diabetes condition, if these acetoacetate and acetone, they also excess in blood as well as in urine. So if the excess amount is acetoacetic acid or beta-hydroxybutyric acid present in the blood, that will lead to ketoacidosis, and that eventually they may go into coma and death. So that's what is the fatty acid, the importance of fatty acid catabolism. Acetoacetyl-CoA, this is again the same mechanism, and then how the fasting or diabetes, how this process is involved. Where you can see the ketone body acetyl-CoA enzyme. This is in mitochondria, and also you can see the brain cell during starvation, how the brain cell using these ketone bodies as a fuel molecule as to produce the energy. Okay, and this is the liver cell, how the liver cells will produce or liver mitochondria will be degrade of fatty acid, and they will produce more of ketone bodies, and that is getting into the blood and blood into the brain cells, and thereby it can act as a fuel molecule for the brain. Okay, and this is exactly this degradation pathway again, where the excess amount of the fatty acids are being released from adipose cells. If you are not eating, that's what glucose is blocked in case of diabetes conditions where the glucose is not used or getting into the cells. Then what will happen, the excess amount of the fat which is stored in the adipose cells, that can be converted to free fatty acids, and they will lead to ketone bodies, blood pH drops, and then you have a coma and death. So that's the process. acetyl coenzyme A and malonyl coenzyme A, how they form with bicarbonate, they can convert to malonyl coenzyme A. Acyl carrier protein, that's what the first step in the synthesis process, the acyl carrier protein ACP, and they also combined with the coenzyme A, and then phosphopantethene group, they were combined to coenzyme A. So now you get principal reaction fatty acid synthesis in bacteria, table 22.2. So how the acetyl coenzyme A, the first step is a bicarbonate ion added to acetyl CoA to with presence of ATP, malonyl CoA, ADP and PI. So acetyl coenzyme A carboxylase enzyme. Then acetyl CoA ACP to form again acetyl ACP plus CoA, and then malonyl CoA plus ACP, malonyl ACP plus CoA. Now acetyl ACP and malonyl ACP combine together to form acetyl ACP plus carbon dioxide. And then acetyl ACP in presence of NADPH and H plus a reduction reaction, beta hydroxy butyryl ACP and NADP plus. And then you get beta hydroxybutyryl ACP that gives crotonyl ACP and water. Crotonyl ACP, NADPH form butyryl ACP and NADP. So imagine you start with the acetyl CoA 2 unit and another acetyl CoA 2 unit to form butyryl ACP. That means 2 plus 2 you get 4. So the enzyme here, the enol reductase. So these are the nutshell. This is the fatty acid synthesis going on in bacteria they have done. And in any other organism also the same procedure or the same metabolism is being followed. Okay. This is again the different steps which is involved, which I explained earlier, and acetyl ACP, malonyl ACP, acetoacetyl ACP, and then hydroxybutyryl ACP. You can see these steps: condensation and reduction, condensation and reduction, and dehydration and a reduction. That's the final step. So these are the 4 steps which is involved. First step condensation, second step reduction, third step dehydration, fourth step a reduction. So this is exactly the opposite of the fatty acid breakdown, but it is the synthesis of fatty acid. And here the selecting and condensing compartment here with the modification compartment. I mean how the different groups, you have the ACP, how the ACP is being added, and these are these mechanisms. This is the endoplasmic reticulum here. How they changes this mechanism, which I am not going through this one. Synthesis of fatty acids, different steps. Acetyl CoA is inside the mitochondria, and this is the cytoplasm. So the degradation process taking place in the mitochondria, but the fatty acid synthesis is taking place in the cytoplasm. Okay, that one should remember. So you can see here, mitochondria, pyruvate, oxalate state, and everything. The citrate is coming in, and then fatty acid synthesis. This is inside the mitochondria, and this is inside the fatty acid synthesis in the cytoplasm. And here the process of the electron transfer. And this is the interesting part with the leukotrienes and lipoxygenase and prostaglandin synthase. And this is prostaglandin synthase with the prostacyclin, other prostaglandin, thromboxanes, and these are some of the product from arachidonic acid. Arachidonic acid is a fatty acid, and this fatty acid will play a major role in controlling inflammation or pain. Whatever we experience in the local area of the tissues, you have the arachidonic acid metabolism is in heightened, is more. So the painkiller, aspirin or whatever the painkiller which you are taking, that has a profound effect in inhibiting the prostaglandin synthase, and thereby you will not suffer from pain. So that's why the painkiller they work on. And we did a lot of work on this for my past several years on this particular mechanism, and they won the Nobel Prize for the prostaglandin synthesis. But in a negative side, these drugs which inhibits the prostaglandin synthesis will give a relief for inflammation, but unfortunately the same drug will produce inflammation in the small intestine. So that's the my work, research earlier. The question is being asked to me when I started my postdoctoral work, how on earth an anti-inflammatory drug induces an inflammation in the intestine. So if you are interested to study why, I would recommend to check my paper in the electronic reserve. You can go into my electronic reserve and then check one other paper, which I can pull it out now. I don't know whether you have done that work before, which I asked you to read some time back. Do you remember, someone? Course home. Oh, here it is, electronic reserve. So that. Okay. Here, electronic reserve, and this is the password you require to type it in. Yeah. So if you go into my electronic reserve and you find there is a paper in think biochemical basis of non-steroidal anti-inflammatory drugs induced damage in the gastrointestinal tract, and a review and a hypothesis. This is the one which we did the hypothesis, and also we proved the hypothesis. It involves both mitochondrial oxidative phosphorylation uncoupling as well as the prostaglandin synthesis inhibition. Both of them are necessary to inhibit, to induce an inflammation. So this is the work and this is a review. I encourage you to study this, and this is the mechanism. Uncoupling of oxidative phosphorylation, could you see from your? Yeah. And the electron transport, this is for the NSAID, the effect of non-steroidal anti-inflammatory drugs that decrease the ATP at increase affecting the cytoskeleton and the substrate flux on HMP shunt pathway that lead to increased intestinal permeability, where you have a mucosal exposure of luminal aggressive factors. And now you can also hear the inhibition of cyclooxygenase. That's how the prostaglandin synthesis. They reduce mucosal prostaglandin, and that will lead to blood flow, cell proliferation, mucus production. That, because of this inhibition, you have a decreased mucosal reparative capacity. That everything put together, you get increased permeability. And then what happens here in the small intestine, you have a proteo and hydrolytic enzyme. Why I am saying this? We are going to deal with the next part of the protein degradation. We are going to study this proteolytic enzyme, hydrolytic enzyme, and ingested foods, bacterial degradation products, pancreatic secretions or bile or bacteria. All of them are increased at the time, the increased permeability, exposure to luminal aggressive factors in the small intestine. And then in the stomach, acid, pepsin, bile, Helicobacter pylori, increasing the neutrophil recruitment. All of them lead to a microscopic biochemical damage to macroscopic ulcer. So this is the mechanism which I put forward in 1992, and the people, they didn't believe at the time. And then after that, we proved this hypothesis with the different experiments, and then they accept this paper in Scandinavian Journal of Gastroenterology and other journal. So this is one of the interesting paper. So you can check here the enzymes, glucose 6-phosphate dehydrogenase enzyme. All these enzymes are 100% here, more than 80%, except the HMP shunt here. Some of the enzymes are 100% present in the small intestinal epithelial cell. So these enzymes already been published. These are the work which has already been published, and these lead to us to hypothesize a new pathway. If you see the glucose, this is the intestinal epithelial cell where the glucose uptake is taking place here, and then this is the glycolytic pathway, and then acetyl coenzyme A which is coming over here. and then the another transporter group 2 which will transport into the blood. So what happened in the epithelial cell, there are the mechanism, a lot of mechanism which is going on, and that really helpful in the lipid absorption or the amino acid transport or glucose transport. So we did a lot of work, and one of the earliest work on the leucine absorption again. This is on the in vitro absorption of the C14 or radioactive leucine. This is one of the earliest one, 1983 paper. That's the pioneer at the time when we did with an experiment where the mucosal to serosal layer of the during inflammatory process, you get a decrease in the absorption of C14 leucine. This is the time course of this, and the result is here. You can see here clearly the normal one, and then this is the inflamed one. You can see this graph, C14 leucine absorption is decreased. The serosal layer is not changed, but mucosal layer, the empty graph is being decreased. And these are some of the anti-inflammatory drugs which we treated. It's become a normal one. So this is but bit ancient one, or the oldest paper which I did. So this provoked me about all the amino acid synthesis, amino acid degradation pathway in later stage. Okay, coming to our point. PowerPoint. This is a prostaglandin synthase and other prostaglandin and thromboxanes, and still people are working on it to have a design a toxic-free, non-sterile, anti-inflammatory drug. So this arachidonic acid metabolism, prostaglandin synthesis, plays a major role in understanding the fatty acid metabolism. So prostaglandin E2, the structure, prostacyclin, another structure, thromboxane A2, another structure. These you can see all of them are derivative of fatty acid. Okay. Active carboxylase, inactive carboxylase with phosphorylation. This is another mechanism how the protein phosphatase will play a major role in carboxylation reaction. Okay, now you can see the electron micrograph of this fatty acid synthesis. And citrate, how the inactive carboxylase become in presence of citrate is partly active carboxylase. That means the citrate is being taken up. So this is some of the dephosphorylated, highly phosphorylated phosphorylase activity or carboxylase activity with citrate concentrations. Phytanic acids. These are some of the experiments which I am not going to go in detail. Okay. With this, I am just finishing it about the fatty acid synthesis and their importance in inflammation or the inflammatory. Oh no, that's not right. Okay. So now I would like to get into the PowerPoint for today's class. That's on the protein or amino acid metabolism. Do you have any questions now so far about the fatty acid synthesis? Any questions? Cinco Ranch? Victoria? So? So now let's go into the next chapter, chapter 23. That's on the protein metabolism or protein turnover and amino acid catabolism. So I want to check with you about how we eat the protein. How do you eat protein? What form do you eat protein? Burger or beans, meat, milk? Yes? Nuts? Yeah, nuts. Yeah. Okay. Any other form? Eggs. Yeah, that's good. Egg albumin. Yeah, egg. Any other form? No? Okay. Milk and milk products and everything go on better. Okay. So, if you eat all this protein and then how the protein is digested in our body. When you eat protein, that protein digestion starts in stomach. Alright? The stomach, what the stomach will do, stomach will secrete, as you know, the hydrochloric acid. Okay, this acid and the protein acidify this protein here. Or in other words, in the acid environment, you have a pepsin enzyme and renin enzyme. Also, this acid will kill the bacteria which is getting into the stomach from the food. So, the hydrochloric acid which is secreted in the stomach will help this protein will be degraded. The protein will be broken down by the enzyme pepsin, renin, this one. And thereby, the complex protein become a simple polypeptide. See that the mono or di, mono di like that, mono means one and di means two, tri means three and poly means many. So many peptide, the protein is made up of many amino carboxy bond, or carboxy we call it as a peptide bond. Meaning, suppose this is the one amino acid, I am just writing the one amino acid here, COHCO, and then here. See, this is the carbon atom, this one group is CR group, another one is H, another one is the nitrogen group, NH the amino group, acid group. So, what you have, this is one amino acid, amino and acid, so we call amino acid. So, this amino acid is joined with the another amino acid of the NH2 here. So, what happens, this is the another amino acid too, okay. So, this is the condensation, this NH2 and water molecule is removed, water is removed, H2O. So, what will happen, the CO and NH join together and thereby we call it as CONH. So, CH2 and then here C, O here, O and then here the NH like, and then this is for the another amino acid. So, this is one amino acid, this is another amino acid. So, they are joined together here. So, this linkage CONH, CO and then this NH, this linkage we call it as a peptide. So, like that there are many peptides which is involved in a protein, like one peptide, another peptide. But in between that you have a amino acid, okay, and then amino acid. So, first amino acid 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, you have a many amino acid joined together and this combination we call it as a peptide. So, the protein, whatever we are eating it's the complex one, So, you have this different structure of this protein and this protein may have this protein may involve directly, may involve directly into the degradation process, and that, I'm just getting back onto it, yeah. So if you have 9 amino acids together, you have 9 different peptides. So this process you have to have a many cleavage, many enzymes to peptidases. The peptides linkage will act by peptidases. So the enzymes we need to cleave these ones. The enzyme we call it peptidases. Peptidases. And I want to check with you, you know, you eat cheese. How the cheese is being produced? Cheese. Huh? You get from milk, and then you add enzyme. That enzyme is renin enzyme. That is from stomach of calf. Baby cow. So they slaughter the baby cow and extract the enzyme from them, and then they add to the milk, and they are producing the cheese. But now the people have vegans, they don't like that meat, and they got vegetarian cheese. How they produce vegetarian cheese? In a way, they used to make like a biotechnology or genetically modified yeast. They produce from yeast. The yeast producing certain enzymes, the same enzyme renin, and thereby are some bacteria, and from that they are getting this enzyme, same renin enzyme, and they used to do it. That's some other things. But initial days, the enzyme, they sacrifice the calf and then getting into the extract more of renin and then add to the milk, and thereby they produce cheese. Why I'm saying this? The renin enzyme, which will also present in our stomach, and what you get the renin, and that will degrade the polypeptide. Whatever the protein which we eat, that will be digested by this renin. Polypeptide become other small peptide. And these small peptides are also, they can also release some amino acids, dipeptides, and they also transform or they transferred from the intestinal lumen. Suppose if you are, you know, it is esophagus, and you are eating it from here, and then this is stomach, and it is getting into the small intestine. So this is what they happen in the renin process, the, you know, hydrochloric acids and everything. And in the small intestine, and here it will be transferred into the blood, and using, you know, at this stage you have a mucosal cells. That cells, they have a transporters. Okay, they have a amino acid transporters, and peptide transporters, So these amino acid and peptide transporters transfer the amino acids and peptides to the blood. So in the blood, what they will do, it will carry on to distribute to the whole body and the tissues. And there again, the cells are the tissues of the body. So now the tissues or cells in the body will resynthesize the protein. Not the same protein, not the milk protein, but the protein of the cells on demand. What type of protein you need, that cell will synthesize. Suppose if the cell is the beta islet of Langerhans of the pancreas, those cells, beta cells, will produce more of the protein of insulin, the protein hormone. That protein they will synthesize. So rest of them, you know, they might able to get remove the amino group. This is the resynthesis of the protein from amino acid. But in the excess of amino acid, what will happen? The amino acid in the excess, unlike glucose, okay, unlike fatty acid, the excess of protein or amino acids, okay, what will happen? If it is unlike glucose, what will happen? If you have excess of glucose, that is being transformed to glycogen. It is storage. Like a fatty acid, if you have excess of fat or fatty acid, that is stored in adipose tissue. So you get it is stored and glucose is stored, but if you excess of protein or amino acid, it is not stored. Not stored. Then what will happen if you have excess of amino acids? If amino acids, that the amino group of amino acid, will be removed, and that will be added to carbonyl group to form urea. So it is excreted as the urea. So excess amino acid, then that is the amino group that is being removed in urea cycle, and then getting into the urine. In the mammal, it's coming in urine. In bird, that is converted to uric acid. In birds. So birds won't urinate, but they have uric acid that give us a paste when it will come out from the fecal matter on that one. But here, in the urea cycle, in the human or the mammalian, you have got the urea cycle is getting into the urine. But this combination of removal of amino group into the urea cycle involve the ATP formation. So this is a way which we are going to deal today about how they are going to convert from amino acid into urea, as well as the rest of the carbon skeleton, the amino group which I said, the urea, and the carbon skeleton of the amino acids, carbon skeleton will go into TCA cycle, and also it can also enter into glucose pathway, gluconeogenesis or glucose synthesis, or also some of them are getting into the fatty acid synthesis. Also they eventually they also produce for the energy. So this is the degradation pathway, and we are going to see how the protein turnover, that's why it is not like protein synthesis in a way, but it is a protein turnover. You are eating in one protein, I'll just write it here, you are eating protein 1 from milk or egg, but that is converted to protein number 2, like a hormone or some other body cellular protein, which is not like just like this, but entirely different. So the process of converting one protein to the another, we call it as protein turnover. I know about apple turnover, but if you want to know about the, apple turnover? You even eat apple turnover? Anyone? Yes, heard of it. You didn't eat? You don't like it? It's not right? Why? You like apple, not apple turnover. Anyone tasted apple turnover in Victoria? Yes? Good. Okay. So what I'll do, I'll give you a break now, and please come back another 10 minutes and we will finish the rest of them. Okay. Let's go again on the, just to summarize what we did on the protein turnover. The degradation of proteins within the cell. Now we are going to start within the cell. See the earlier version which I discussed earlier, from whatever you are eating it, that protein is being digested by digestive enzyme from the stomach, and then as well as it's getting in the small intestine, and then it is being transferred into the cells and the blood, and blood to the different tissues. And the tissues, those peptide or those amino acids is being consumed or converted into their own protein, their own product, their own hormones or enzyme or any other cellular material. That's what we did. Now we are going to study the degradation of proteins within the cells. As I mentioned earlier, if there is a cell and there is a nucleus and they have the cellular membrane, and the membrane also have a protein, and the nuclear membrane also have protein. Cytoplasm, they have a lysosome, Golgi, and different types of proteins which is hanging around in the cytosol as well. So we are going to study how on these protein or the membrane protein or the cytosolic protein or the nuclear protein or the nuclear membrane protein or the endoplasmic reticulum, and those proteins being degraded within the cell. That's what we are going to see now. Okay. Right. The cellular pathway. Okay. The steady state supply, actually supply of amino acid to the cells. Whatever we did study before, degrade the protein from the digestive system, protein. They provide the steady state supply, of what? Amino acids. And to the cells, and tissues. And they synthesize their own protein. But these cellular protein are constantly degraded. They are constantly degraded. Degraded, one word, constantly degraded. Why they have to degrade? Now my question is, why the cell, the protein which is present inside the cell degrades? Why the degradation? Number one, if the cell get a damage or inflammation, or the cell is not behaving properly, or cellular behavior in a way that is in the immunological cell, immune cell. They are supposed to act on bacterial or viral, but it is not. That means that cell is not likely to be there, and that should be executed. So there are certain type of cells will engulf the cell, like for example macrophage, and as well as the white cell or neutrophil, okay, and the white blood cell, leukocytes and neutrophil, and they actually phagocytosis, meaning they swallow the bacteria or the virus or some damaged cell itself, For example, in the cancer cell, the T cell, and they call it as a T killer cell. And these cells are the immune cell, thymus derived, the killer cell, T killer cells are the T cells, and they can act on cancer cells. Suppose the T cell, this is a kind of white blood cell, and if they encounter a cancer cell, cancer stem cell or cancer cells, and it will recognize it is not belonging to us, so what? They will attack on this cancer cell, and thereby they reduce the cancer cell, and they are called it as a cytotoxic T cells. Cytotoxic T cells. And if you increase the number of cytotoxic T cell in your body, in that case you can eliminate cancer. The cancer cure, you can do it. How of these cells, cytotoxic T cell, are inside any cell, how the protein is degraded, that's the part which we want to look into that right now. Okay. So the suppose if the protein is misfolding, meaning why we have the folding? As I mentioned earlier, suppose if this is the one amino acid, if you go on straight line, there's a one type of structure. Another type of amino acid, this is amino acid 1, amino acid 2, amino acid 3, amino acid 4, amino acid 5, 6, 7, 8, 9, 10. So they are very meticulously arranged in a straight line. So that is a type of structure of a protein. Another type of structure is something like this protein will come like this and then zigzag, 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 zigzag. So the arrangement of one amino acid here, another amino acid here, another amino acid here, another amino acid, another amino acid, another amino acid, another amino. This is they call it the alpha sheets or these are beta sheets of this arrangement of the chain or the peptide chain or this one. In other words, what will happen? The combination of the primary amino acid sequence and this is the secondary structure, beta sheets, secondary structure, and they form, combined together. They can also for tertiary structure, meaning some of them are straight and some of them are folding like this, and some of them are straight and some of them are folding like this. This is the way they can constitute. A protein can constitute a straight line, a zigzag, zigzag, and then a straight line, and then a zigzag. So if a combination of all these three things together, we call tertiary structure. In other words, they are folding. And this folding is necessary for the active site. Suppose this is the enzyme molecule, also a protein. They have got some sort of active sites or the binding site. Suppose this is the binding site, this is another binding site, this is another binding. This is one enzyme, one molecule made up of protein, but they have a particular fashion of folding. And this folding is necessary for the enzyme activity. In case of mutation, what will happen if this folding is not right? Suppose if this and you have a misfolding, suppose like this. This is a misfolding. So you cannot have, this is a wrong protein. The protein is not folding properly. It's a misfolding protein. Misfolding protein. And this is not necessary for the cell. If it is damaging the cell, and the cell will execute it, this should be degraded. For example, one of the misfolding protein which is present when you eat a mad cow disease burger, that is getting in your, what type of disease is that one? Your brain, yeah, brain, that's correct, yeah. The brain is damaged on that one because of the misfolding of this protein. What type of things that disease? You call it a mad cow disease? Yes, that's what for a long time ago. So this is happening. The misfolding and correct folding, it is constantly happening in our cell, and those folding of those proteins should be removed from the cells, from the damaged cells, as well as the cancer cell or any other diseased cell or bacterial or any other thing. It should be removed, and these unwanted cells, in other words, unwanted, they are not needed, unwanted, and they are not cycled, uncycled. These proteins are not, or amino acid is not being useful. Uncycled protein, this protein is not useful or recycled, as I mentioned earlier. They are scavenged by, they are scavenging, meaning something is attached to it. In other words, they are ubiquitinated. Ubiquitinated means there are another molecule like a ubiquitin molecule, and that molecule is attached. So suppose this is the unwanted protein, this ubiquitin will attach to it. Uncycled protein, the ubiquitin attached to it, and thereby they tag them up. These are the unwanted material, these are the recycled, uncycled, this is not used, unused protein, unwanted protein, and that is being added with ubiquitinated. This is another term which you should remember. Ubiquitinating a protein means that protein is ready to die, ready to execute. In other words, if you want to remove the protein, you have to identify the protein by ubiquitination, and thereby the ubiquitinated the protein like this. Suppose these are the ubiquitinated protein, these are the ubiquitinated protein, these are the ubiquitinated protein. These are the protein, unwanted protein by ubiquitinated, and they have to undergo, a degradation. So this protein is tagged, they are identified. Now this protein is going to be executed in proteasome, and these proteasomes are present in each and every cells. All cells, they have the proteosomes, and these proteosomes will suppose these are the arrangement of proteosomes. I'll show you in the picture. But they have a series of proteolytic enzymes which is attached inside when the protein, ubiquitinated protein only, not all the protein, only the ubiquitinated protein will pass through, it will be degraded. It will be degraded because of the enzyme which is present inside the proteasome, and this is proteasome. Proteasome. And this is something like a ribosome, nucleosome. This is a proteasome, and proteasome will degrade all the ubiquitinated protein, which is unwanted protein, and thereby which will increase more of these amino acids being removed from the cells, or the unwanted protein is removed from the system. So for this process, you have to add ATP. You need the energy process, ATP. Okay, proteasome is now the role of proteasome in cancer biology. Proteasome inhibitors will act as another therapeutic index in the cancer cell, because if unnecessarily the proteasome is highly activated in cancer cell, even the normal protein, even the normal useful protein is also ubiquitinated and thereby degraded, and what happens, the cancer will proliferate. More of cancer cells is formed because of the highly activated of proteasome. So people now will think they will add chemotherapy plus proteasome inhibitor together, and thereby they can stop the cancer proliferation process. Why I'm saying this? Because I did work on the proteasome inhibitor and chemotherapy in a clinical trial, the experiment which I did my research in the cancer biology in UNC Chapel Hill, University of North Carolina, where we did a proteasome inhibitor 341 with Millennium Pharmaceutical they have supplied, and then we have added the proteasome inhibitor and chemotherapy to the mice, and thereby we can be able to see the cancer is tremendously, when compared to chemo alone, proteasome inhibitor, they inhibit the cancer proliferation. That's a very remarkable studies, and we published some work as well. So proteasome inhibitor that will act as a cancer cell, cancer chemotherapy. So in your context, how the role of proteasome is it's very important. We will see in the slideshow. So why we have to do it in this protein turnover in this chapter? Because the protein degradation by proteasome, the excess of amino acid, that should be used as a metabolic fuel. Metabolic fuel. How this amino acid is getting into amino group? Amino group. The amino group will go through urea cycle. You will see that one. And the carbon one will get into glucose. Glycolysis or glucose synthesis will go in that, or fatty acid synthesis as well, and thereby it can recycle. Okay, the carbon skeleton, acetyl-CoA, and then acetoacetyl-CoA, pyruvate, and then TCA cycle, and then can also produce energy. Okay, and the also involved the amino acids, the product of this particular amino acid can also form the pyridoxal phosphate. Pyridoxal, P-Y-R-I-D-O-X-L, pyridoxal phosphate, and they are the coenzymes. They can also act as coenzymes. In also, in the genetic disorder or the inborn errors of metabolism. Have you ever heard of inborn errors of metabolism? Inborn errors of metabolism. One example is phenylketonuria. Phenylketonuria. In the phenylalanine content, if you see the artificial sweeteners, you will find phenylketonuric. It's a warning because it contains phenylalanine. Because the persons who has the inborn errors of metabolism in the phenylketonuria, this is a case where the phenylalanine is being converted to tyrosine. This is a normal process. But this enzyme, phenylalanine tyrosine enzyme, the conversion is being blocked for this phenylketonuria. So they cannot digest the phenylalanine into tyrosine. So they will convert the alanine into the transamination process to form phenylketone, and they are excreted in the urine. So it is called phenylketonuria. So that the disease, inborn errors of metabolism. To check this one, what you need to do, you have to avoid phenylalanine food. Phenylalanine contained food you have to avoid. Otherwise you will get this, and that is lead to lethal process. So like this, there are several inborn errors of metabolism in the amino acids pathway, in the synthesis as well as the catabolism pathway too. Okay, so you have to avoid those. So now I would like to get back onto the PowerPoint, and let me see, protein turnover. Now you can see, I mean, this is your textbook, which is the amino acid catabolism. Getting in the arginine, because I'm going through the TCS cycle, where arginine succinate into the arginine, and this is the urea cycle, by the way. And the urea is removed from arginine, and then ornithine, and ornithine acetylene, and this pathway is going on. And this where the urea cycle is present? Urea cycle is present in liver. Please take down the notes. Urea cycle is present in liver. Okay. It is not in the kidney. Most people, they write urea cycle, urine, oh, it's in the kidney. So that is wrong answer. And urea cycle is present in the liver. Okay. How the urea is formed? That's typical questions in a major in biochemistry students, and that's the urea cycle. For the determination of urea cycle, a person got Nobel Prize for the urea cycle, because that's the key pathway which is involved in the removal of amino group. And the first group is the removal of deamination reaction, and the more ammonia, if your blood system is get excess ammonia toxicity, because the urea cycle is not operating, what will happen? You have excess of ammonia, that will lead to retard into your brain, and it will shut off the brain, and you will go to die immediately. So ammonium, that's a toxic ammonium. And this is the carbon dioxide as well, plus ammonia to form carbamyl phosphate. Both of them are unnecessary. This is a excretory product for our system. So it enters into this cycle, urea cycle. And these are essential amino acids in human beings. So you should remember, you have to take the essential amino acids, meaning this amino acid is not being recycled, which I mentioned earlier. Our amino acids are, whatever the protein is recycled and resynthesized in our own body. But the essential amino acids are not synthesized in our system. We have to depend upon external food or dietary resources. So that by we can, we can maintain our protein metabolism in our body. If you are not taking the histidine or isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine, what will happen? You are going to suffer protein malnutrition. So even if you take other protein, if the protein is not having these amino acids, then you are going to suffer a malnutrition, protein malnutrition. So what the people normally do, they add these amino acids externally, or the protein contain excess of the lysine, leucine, isoleucine. You have to eat more of this so that keep your protein balance. Okay, these are what is the essential amino acid, what is non-essential amino acid. You should remember. Okay, that's what I'm saying now here. Proteins, proteolytic enzymes, how they degraded, which I mentioned earlier, amino acids, oligopeptide, and then is getting aminopeptidases. It will remove amino group. Okay, and then it is intestinal cell, the transporter, the green and red are the transporting proteins which will transfer from the lumen, whatever you eat, which is the amino acids, and that is going to be transferred into the cell with the amino acids and tripeptide, dipeptide, and inside the cell, peptidases, intracellular intestinal peptidases. They will be activating and converted to amino acids, and these amino acids are getting into the blood. So from the blood to the tissues. So that is the cycle. Okay. Now, what happened, which I mentioned earlier, ubiquitin. So these are the ubiquitin molecule. These are C terminus or this one amino acids. How the ubiquitin molecule is surrounded here. So the stick model, which is the protein, and the ubiquitin is going around that protein, so that it capture everything, and thereby it will go through the ubiquitin molecules. You can see the ubiquitin, and getting into the, say, this ubiquitinated, and these are the isopeptide bond. How the ubiquitinated compound are getting into the proteasome. These are the lysine amino acid, and this is structure of the amino acid. Another amino acid, this is the peptide bond, which I explained earlier in our class. Then these are the ubiquitinated with the ATP, ubiquitinated AMP, and then enzyme AMP is released, and now ubiquitinated is added to E1, and then you go on to the next step. Next step is the target. Now the next step, and then this is the target where this ubiquitinated is being, it is the ubiquitin, and this is the ubiquitinated protein. This is the target protein. The yellow one is the target protein, and this is the ubiquitin is activated with the ATP on these steps. When it is activated, it can the enzyme 2, and then it is being with the enzyme 3, and this is a target protein. Now what happened? It is going to degrade the target protein. The two target protein, you can see this one, how the ubiquitinated working, the mechanism, how the isopeptide bonds. And these are some of the half-lives of cytoplasmic yeast protein. One of the experiments they did with highly stabilized residues, intrinsically destabilizing residues, and they have done the mechanism of action. And you can see the alpha subunit, beta subunit. This is the proteasome, structure of proteasome. Okay, alpha subunit, beta subunit, alpha subunit. They are inverted, these two subunits, so that you can have a hole in the middle. And if you have the protein getting in, and that is going to be like a scissors with a proteolytic enzyme that is be degraded. This is the proteasome structure. This is the N-terminal threonine nucleophile, where exactly how the amino acid is binding to the proteasome enzymes inside. You can see this cap, catalytic core. So these are the cap, and this is the catalytic core, and this is the cap. So this is the yellow area where the cutting is happening inside the proteasome. This is inside the proteasome again. You can see the ubiquitinated protein. You can see these are the ubiquitinated. The blue one is the protein, polypeptide. Getting in, what happened here, peptide is fragmented, large proteins fragmented, and proteolysis, amino acids is coming up. And then here the released ubiquitin is go back onto this, and then ubiquitinated again. So these amino acids left intact for biosynthesis. These amino acids will go into useful other cellular protein biosynthesis. That's a recycling. But if the deamination progress, amino groups enter into nitrogen disposal cycle, that is the urea cycle. And then the carbon skeleton of the amino group will enter into glucose or glycogen synthesis, fatty acid synthesis, cellular respiration, that can also produce energy. And these are the pretty much of a protein degradation pathway. And let us see, this is in detail account on this PowerPoint. And you can see the archaeal proteasome and eukaryotic proteasome. There's a comparison, different organisms. And this is the drug which I want to explain to you, bortezomib, a dipeptidyl boronic acid, and this is a proteasome inhibitor and an excellent candidate for the cancer, especially multiple myeloma and some of the melanoma cancer. And they are also trying for the breast cancer, different cancer. The proteasome is heightened activity, and this is a successful drug, and it is in the clinical trial, and now it is coming into the market. This is called the bortezomib, and everybody is talking about this drug is a miracle drug for the cancer therapy. Not only that, it will act along with the chemo as well, and it will also prevent chemo resistant. So that's what the promising news of that particular drug. So this is again the mechanism of how the amino acid is getting into thymine, the biosynthesis of nucleic acids. Process regulated by protein degradation. What are the processes regulated protein degradation? So if the protein degraded into amino acids, it can enter into gene transcription process, cell cycle progression process, organ formation process, circadian rhythm process, inflammatory response, tumor suppression, cholesterol metabolism, antigen processing. So the antigen processing is the one where we did a study in immunology class, where the bacterial peptide or viral protein which is being processed by the same mechanism of the protein degradation mechanism, and thereby the antigen processed and then antigen presenting cells into memory cells which will synthesize more of antibody against this particular antigen, or antibody against the virus, or antibody produced against the bacteria, and thereby we can boost the immune system. But in all of these processes, you need to get the protein degradation. That pathway should be active. This is again the same mechanism of the ubiquitinator, and then how this ubiquitinator will act again. This is again the mechanism again. Amino acids to glutamate. This is another pathway how the amino acid is being converted to, the amino group. The blue is converted to a urea. So amino acid always combined with a glutamate, converted to glutamate, and glutamate to get an ammonia. So glutamine deaminase reaction, more of ammonia is formed. Now what happened to this ammonia? Aminotransferase, either that will transferase here, another mechanism of glutamate, how the glutamate is releasing this ammonia, that is the mechanism here. Shift base and then ammonia is released, alpha ketoglutarate being produced, and this ammonia is now where that ammonia will go now. Again the same process, amino acid to keto acid, and the glutamic, alpha ketoglutarate to glutamic, and glutamate to alpha ketoglutarate. This is a cycle, and the NAD plus H2O, NADH plus ammonia, and now this ammonia is being converted to urea. How? The urea cycle, let us see that one. During this process we have studied pyridoxine, vitamin B6 synthesis, pyridoxal phosphate PLP, and the PLP donator, and how phenolate. This is the mechanism of the coenzyme, the ammonium transfer reaction. Again this mechanism of coenzyme. Okay, ketamine, now the ammonia is released. Pyridoxal phosphate PMPE. Again all these mechanisms are there. How this particular pyridoxal phosphate is being released. Bond most perpendicular delocalized pi orbital. This again the mechanism how the molecule is releasing this ammonia, okay, of the cellular structure. Aspartate aminotransferase, serine hydroxymethyl transferase enzyme. Bond most nearly perpendicular to the p orbital. So the bond which is perpendicular p orbital, they're more vulnerable, they're easy to transmutate. So now here the serine, and then got the amino acrylate, ammonia is released, pyruvate. Okay, this is again from amino acrylate to ammonia, how the ammonia is being produced. Now the liver, glutamate to ammonia, and the ammonia into urea, that's on the liver pathway. In the muscle, glucose to glycogen to glucose, glucose to pyruvate, alanine getting into the liver, and then ammonia is being produced. So in all these cases here, the branched chain amino acid especially present in the muscle, that will also produce a carbon skeleton for the cellular respiration. The ammonia is removed, and that is converted from pyruvate to alanine using this branched chain amino group to form alanine, and alanine getting into the liver and getting urea cycle. In all these cases, final end for the ammonia is the urea cycle. Okay. Now here the ammonia getting into the urea. See, COO, C here the carbon, C double bond O, NH2, NH2. This is the structural formula of urea. Now you can see this is the urea cycle, how the urea cycle is linked now with the carbon dioxide, ammonia put together, carbamyl phosphate, carboamyl phosphate, and this carbamyl phosphate is present in the mitochondrial matrix, because mitochondria is the site where the ammonia is diffused, as well as you get the carbon dioxide in the matrix. They form the carbamyl phosphate, and citrulline is being produced inside the matrix, and citrulline is coming out into the cytoplasm arginosuccinate with the aspartate molecule, and then fumarate is released because aspartate entry here, argininosuccinate fumarate is released, and arginine. And arginine with water, very easy. With water, the arginine will release urea. So that's what the urea is synthesized in liver. And then the product, side product is ornithine, and the ornithine with another molecule of carbamyl phosphate getting into citrulline pathway. So this is a cyclic process going on. More and more carbon dioxide, more and more ammonia is being removed in the form of urea. So that is the net reaction. I mean, how the bicarbonate, the carbon dioxide which is present in the blood, and how that is being converted into carboxyphosphate. And then ammonia to carbamic acid is synthesis from ammonia, or the release of inorganic phosphate. With here ATP, another 2 molecules of ATP you have to spend in order to produce the carbamyl phosphate. This is the energy requiring process. Okay. Now the carbamyl phosphate with ornithine, this is the ornithine molecule, carbamyl phosphate getting into ornithine trans carbamylase reaction, where it will produce more of citrulline, C double bond O NH2 here, okay, and NH here. So when it cleave here, CO NH2, so the urea will be released, but it requires arginine. It's not directly going into that pathway. So citrulline now with aspartic acid, aspartate, and this is also another amino acid which combine together to form the ATP and the AMP arginosuccinate synthetase, and you get arginosuccinate, and then here this is your CONH2 group over here. And now we can go into the arginosuccinate to arginine. So here the arginosuccinase is enzyme, arginine is produced, and fumarate is being released. And then the fumarate will also enter into the TCA cycle, and the arginine now will release with the hydrolysis, will remove this urea, and it will produce ornithine. So you can see this one in this one, arginase is enzyme with water, ornithine, and urea now is being produced. So thus we explain how the ammonia group is removed from amino acid and form urea in liver, and using an urea cycle, and part of it also shared with the TCA cycle enzyme with the fumarate and succinate enzyme. Now you can see in this one here, fumarate, malate, oxaloacetate, aspartate. So fumarate, malate, oxalate, and this is a part of what? Of TCA cycle. And arginosuccinate, citrulline, ornithine, arginine, this is a urea cycle, where they meet each other, argininosuccinate. So you have to be remember, oxaloacetate can also give rise to glucose and the gluconeogenesis pathway. So in amino acids degraded, it can also get into glucose or get into the TCA cycle, or also get in the ammonia is removed in the urea cycle. So you have to be remember in all the mechanism which is involved in this urea cycle. Let us see. N-acetyl glutamate. This is again aspartate fumarate mechanism of action with the coenzyme. How the citrulline is produced, and arginosuccinate. This is again the benzoate, benzoyl-CoA, hippurate, and excretator for the glycine and the ammonia. This is another excretory product. Phenylacetate, glutamine is excreted. These are the how the drug is being excreted. If you are taking excess of benzoate or benzoic acid, excess of drug is catabolized, and thereby it's being removed, and then it's excreted in urine. And here you see different amino acid, alanine, cysteine, glycine, threonine, tryptophan, entry pathway, pyruvate, which will degrade all of the removal of ammonia group, and then the carbon skeletal group entered through the pyruvate, and pyruvate into acetyl-CoA for fatty acid synthesis, or pyruvate enter into the oxaloacetate to get into the glucose pathway, or it can enter into TCA cycle. So aspargine enter, aspartate enter oxaloacetate, aspartate phenylalanine tyrosine enter fumarate, isoleucine amino acid methionine threonine valine, they enter into succinyl coenzyme A. So in the intermediary metabolism, all these protein is degraded into different amino acids, and each amino acid is having separate entry into TCA cycle, and this is the net of the sum of all these amino acids, where they enter and how they are involved, either it produce glucose or produce energy or produce fat. So that one should remember in the fatty in the amino acid degradation and the fate of amino acids, individual amino acids. If I ask a question, what is the fate of glutamine in the protein degradation pathway, then you have to say glutamine is entering into alpha ketoglutarate and thereby is getting TCA cycle. Suppose if I say lysine, where the lysine will enter, you can also say lysine can enter into acetoacetyl coenzyme A. Okay, and this is a reversible pathway, and then can enter acetyl CoA and then TCA cycle. Okay, if I say threonine, and you should say threonine will enter after the getting succinyl coenzyme A, and tyrosine fumarate. Okay, so you should be very careful. So suppose if the aspartate can enter oxaloacetate as well as aspartate getting into the fumarate. Okay, so the tyrosine can also enter fumarate, and tyrosine can also enter into acetoacetyl coenzyme A. So it's it's all of a different pathway where it can enter into TCA cycle. So these are the individual amino acids, how they enter into the pathway. Proline and histidine, and then glutamate and proline, arginine, glutamate, all of them. Propenyl CoA, methylamine CoA, succinyl CoA, and methionine. These are sulfur-containing amino acid. How they enter homocysteine, and then getting into succinyl CoA. If I say methionine, what is the metabolic pathway of methionine? You have to say this S-adenosyl methionine are SAM and S-adenosyl homocysteine, homocysteine cystathionine, alpha ketobutyrate and propenyl CoA to succinyl CoA. So you have to have a large number of steps which is involved. So normally the people, they ask the question, what is the fate of methionine? Though you have to say all these steps till succinyl CoA. Okay. What is the fate of leucine? Then you have to write isovaleric CoA. These are the steps you have to write in your exam. Okay. Now beta-amyl acetoacetyl CoA. Phenylalanine. What is the phenyl? This is Dr. G's research of tetrahydrobiopterin phenyl hydroxylase. He has done extensive work on this one, and then he has also published some work, I think probably in your biochemistry class and other classes. Dr. G must have discussed with his research of phenylalanine tyrosine hydroxylase enzyme, tyrosine synthesis. I think you might have remembered that. So these are the dihydrobiopterin, tetrahydrobiopterin and quinoid dihydrobiopterin. These are the mechanism how the conversion from phenylalanine to tyrosine is taking place. Okay, phenylalanine, tyrosine, homocysteine, methylamine, fumarate, and acetoacetate. These are the degradative pathway of phenylalanine. If I ask a question, what is the fate of phenylalanine? You have to say phenylalanine, tyrosine, para-hydroxyphenyl pyruvate, homogentisate, 4-methyl, 4-mallyl lactoacetate, 4-fumaryl acetoacetate, and fumarate and acetoacetate. So finally end product acetoacetate. Tryptophan. If you say tryptophan, what is the product of the tryptophan, or what is the pathway which I asked the question? You have to follow through this one. N-formylkynurenine and kynurenine, and then 3-hydroxykynurenine, and then 3-hydroxyl anthranilate, and 2-amino 3-carboxymuconate, 6-semialdehyde, and another 11 steps enter into acetoacetate. So you should be very careful on tryptophan. And these are the homogentisate air and highly colored polymer, which is coming onto the phenylketonuria acids, where the urine becomes darkened. Another one is the dinitrophenylhydrazine with the hydro derivative. So these are some of the amino acid. This is the mechanism of phenylalanine mechanism. So inborn errors of metabolism in the amino acid metabolism, where you will find citrullinemia, where excess of citrulline is present in the blood, arginosuccinate lysase enzyme deficiency. If I ask a question,
give me a disease where the enzyme, tyrosine degradation enzyme, you have to say tyrosinemia. Or give me a disease which is the deficiency of tyrosinase, or the mutation or the malfunction of this. Albinism, that means you don't get pigment, melanin pigment, so the person will become albino, okay? And homocystinuria, another disease. Hyperlysinemia, another disease. So you can see the symptoms. So this is a very good question for your exam, like a match the following or something like that. So please remember this table. And this is again the different structural identification here. Amino group, but there is no amino group here. A and B. And C, D, this reaction compound A. And how the proteasome inhibition, which is different step, where the ATP is required. So a percentage of protein is substrate is hydrolyzed, you need ATP to degrade this protein, whereas here it is not. So you have to have ATP, and then only the proteasome is being activated. So that is even another example of this. And proteasome plus ATP, proteasome pan plus ATP, you can see that one here. So this is the confirmatory experiment with the protein degradation pathway. Okay. Any questions? From Victoria. Cinco Ranch.
No questions.
No questions? That's wonderful. In Sugarland? No? Partly I explained to the different steps which is involved, but you should remember the name of the product and names of the enzymes in different amino acid degradation pathway. Okay? If you remember that, that is pretty much we required for your exam. I don't want to get into details of how the conversion is taking place by the mechanism or with the coenzyme A. That is not required for you as a biology students, and that is mainly for the biochemistry majors. And I'm not going into that detail. And the next class, what we need to do, biosynthesis of amino acids. And then we need to have a couple of classes, right? You have to have a nucleic acids, and then I expect you to study some of the research paper, which I will go through now.